Before discussing how the immune system reacts to SARS-CoV-2, we must first know how this virus enters the body and replicates. SARS-CoV-2 is a single-strand RNA virus surrounded by spike proteins that bind to receptors on the cell membrane. When the virus proteins bind to the cell receptors, the cell engulfs the virus through endocytosis. The virus then uses the host cell ribosomes to translate its RNA genome into viral proteins for each part of the SARS-CoV-2 virus, including the spike proteins, nucleocapsid, envelope, and membrane. The parts of the virus assemble in the host cell and exit through exocytosis, with each virus going on to infect a new host and repeat the replication process. The innate immune system is the first line of defense against viruses like SARS-CoV-2 and other foreign substances. It includes physical barriers, secretions, cellular defenses, antimicrobial proteins, and inflammatory responses. Physical barriers like skin and other epithelial cells prevent entry for foreign substances. Secretions can either wash surfaces or create hostile environments. Mucus in the digestive, respiratory, and urogenital systems, tears, urine, and saliva cleanse areas of the body and wash away substances that could be harmful to the body. Secretions that create hostile environments include those that produce lysozymes that digest bacterial cell walls or others that have a low pH. Cellular defenses are cells that detect non-self cells, called phagocytic cells. TLRs help phagocytic cells identify non-self pathogens. The cellular defenses that attack SARS-CoV-2 are natural killer or NK cells, which identify virally infected cells and induce apoptosis. Antimicrobial proteins are part of the complement system, which causes reactions that lyse cells infected with the virus, such as SARS-CoV-2. Infected cells release interferons, which limit the transfer of the virus to healthy cells. Inflammatory response, another innate immune response, increases blood flow, dilates blood vessels, increases blood vessel permeability, and often increases temperature in an area. Mast cells release histamines, which create inflammatory responses. Cytokines are a phagocytic cell that also activate inflammatory responses and recruit other immune cells to create this response.
When the SARS-CoV-2 virus enters a person's body for the first time, the pathogen is foreign to that person's immune system, triggering the primary response by adaptive immunity. The primary response usually takes effect within 10 to 17 days after the initial exposure and is a longer response compared to the shorter, more efficient secondary response. Once the pathogen enters the body for the first time, there are two different responses that occur depending upon the location of SARS-CoV-2 in the body: the cell-mediated response and the humoral response. The cell-mediated response occurs when SARS-CoV-2 has already infected the cell. This host cell will send out signals to T cells so that action can be taken against the pathogen. The T cells will now undergo clonal selection, where the cells will divide and differentiate into effector cells and memory cells. Clonal selection occurs in both B and T cells through the binding of an antigen to a mature lymphocyte. With the T cells, the effector cells are short-lived cells that serve as the immediate response to an infection, including helper T cells and cytotoxic T cells. Once activated by an antigen, helper T cells function secreting cytokines that stimulate other lymphocytes. Cytotoxic cells function in secreting proteins to destroy the infected target cell. Now that the host cell has been infected with SARS-CoV-2, the pathogen will release antigen fragments. These antigen fragments will bind to an MHC molecule, or major histocompatibility complex, which is then displayed on the cell's surface. The cytotoxic T cells are then able to recognize this host cell as pathogenic and take action. The T cell antigen receptor binds to the antigen fragment displayed. Once attached, the T cells secrete enzymes to destroy the host cell and the virus along with it. The other T cells, the memory T cells, then remain in different lymphoid organs such as the lymph nodes, thymus, spleen, and bone marrow, awaiting the secondary response. Another immune response is a humoral response, which occurs prior to SARS-CoV-2 entering a cell, when the pathogen is floating around that person's bodily fluids. In this case, a body utilizes B cells as well as helper T cells. B cells are found constantly patrolling the bloodstream until they receive a signal from a helper T cell that there is a pathogen. These B cells are covered in Y-shaped antigen receptors that contain 4 polypeptide chains, 2 heavy chains and 2 light chains linked by disulfide bridges. The receptor is made up of constant and variable regions. The variable region serves as the antigen binding site, as its amino acid sequences varies depending on the type of B cell. The constant region has little variation of amino acid sequence and determines the mechanism for destroying the antigen. These antigen receptors on the B cells will recognize the antigen on the SARS-CoV-2 and bind to the epitope. This triggers the B cells to produce cells that secrete antibodies, or immunoglobulin, an identical and soluble form of the epitope of the original B cell antigen receptor without the membrane anchor. The antibodies will then latch onto the epitope of SARS-CoV-2 and directly attack the pathogens in the body fluids. Antigens of SARS-CoV-2 can also be found independent of the pathogen, in which the antibodies undergo the same process for destroying the pathogen. With the help of the antibodies from B cells and the cytotoxic T cells, SARS-CoV-2 is now eliminated from that person's body, awaiting future infection.
After the person recovers from having SARS-CoV-2, he or she will have an immunological memory, a long-term protection. In the primary immune response, the exposure to the virus changed the speed, strength, and duration of the immune response. Now that the person encountered the virus again, there will be a secondary immune response. The long-lived memory cells from the primary response to SARS-CoV-2 is what we can attribute the faster response to. These long-lived memory cells are the T and B cells. These cells will recognize the virus and create thousands of effector cells. This process begins in the lymph nodes, where lymphocytes with antigen receptors and epitopes, the part of an antigen molecule to which an antibody attaches itself, will match, activating the lymphocyte bearing the receptor. In this case, the antigen is the spike protein. Now the B or T cell will undergo cell division, creating identical cells. Some of the clones become effector cells, which attack the antigen immediately. The effector form of a B cell is a plasma cell, which secretes antibodies. The effector forms for T cells are helper T cells and cytotoxic T cells. The rest of the cells that didn't return to B and T cells remain as memory cells in case the body runs into the same antigen again and needs to repeat this entire process over again. After these clones are created, the new B and T lymphocytes are now ready to fight. Their defenses can be separated into two categories: humoral immune response and cell-mediated immune response. The humoral immune response occurs in the blood and lymph. Here, antibodies neutralize or eliminate toxins and pathogens. This is considered active immunity. In the cell-mediated immune response, the specialized T cells destroy the actual host cells infected with SARS-CoV-2. In general, they can also defend against cancer cells and transplanted tissues. T cells relate back to innate immunity, as the antigen will be recognized by major histocompatibility molecules, which are host proteins that provide antigen presentation.
The different parts of the immune system allow the body and cells to protect themselves from foreign substances that could potentially be dangerous. Although it is not always effective, it protects us from many forms of harm that could be deadly if the immune system did not complete its intended purpose.